Picture a seasoned executive navigating a sudden market crash. Instead of pivoting, they freeze, snap at their team, and double down on a failing strategy. This executive dysfunction is a biological hijacking of the nervous system. The human brain contains roughly 86 billion neurons, generating the immense processing power required for executive decision-making. Leadership relies on a delicate tug-of-war between two specific regions: the frontal lobe, which handles logic and executive function, and the limbic system, which controls primitive emotions and survival instincts. When an organization encounters intense stress or sudden change, the brain interprets the corporate crisis as a physical, mortal threat. In response, the frontal lobe actively shuts down. All operational control is handed over to the limbic system. This neurological shift floods the body with a massive spike in cortisol, the primary stress hormone. In this state of low brain coherence, communication between different brain areas breaks down. Decisions are driven by primitive denial and anger, rather than strategic problem-solving. How do successful leaders bypass this hardwired biological panic to guide their teams effectively? They actively regulate their hormone levels. This definitions table isolates the specific scientific links between our internal chemistry and our external leadership behaviors. Effective leadership requires suppressing cortisol to manage stress, while simultaneously elevating testosterone to promote confidence and decisive action. Leaders achieve this balance through deliberate physical and psychological routines. They anchor themselves in the present moment, exercise, adopt power poses, and consciously shift their language patterns to regulate these chemical levels. Cognitive leadership relies on this hormonal baseline. Without it, the frontal lobe remains functionally inaccessible. Once that biological equilibrium is restored, leaders can access a target state defined in this academic chart as the three brains of leadership. First, the head brain handles thinking, analysis, and language, driving an organization's creativity. Next, the heart brain processes emotional priorities and generates compassion. Finally, the gut brain acts as the core of identity, fueling the courage and will to act. A highly effective leader integrates all three centers. This restores complete brain coherence, creating a foundation for powerful and balanced execution. Biology is contagious. A leader's internal neurological state broadcasts to every person in the room. As shown in this terminology chart, humans possess special cells called mirror neurons. These make us inherently social and highly sensitive to the emotional states of our peers. If a leader remains trapped in a high-stress, low-coherent state, they involuntarily transmit negative emotions and narrowed attention to staff via mirror neurons. This lecture summary table outlines how teams react to that transmitted stress. They inevitably progress through 4 stages, starting with denial and anger, before eventually moving toward exploration and acceptance. Guiding a workforce through these stages requires an environment that neutralizes the brain's collective threat response. Because the brain perceives ambiguity as danger, the leader must provide external safety signals to quiet the limbic system. This is achieved using the 7 C's of change management: clarity, continuity, certainty, consistency, cooperation, confidence, and communication. These concepts act as precise environmental cues. Factors like consistency and clarity directly lower the group's collective cortisol levels by replacing neurological uncertainty with predictable data, triggering wide-scale coherence. Organizational success is rooted in this biological reality. Leadership effectiveness is ultimately measured by how well a leader manages the human nervous system to drive collective achievement.